Welcome to the Bio 101 channel. My name is Samantha Chesterfield, and today we have Ellen Valencia, a professor from the University of Hartford. Accompanying her are her two students, Shaniqua and LaQuisha LeBron, the LeBron sisters. Come on in. Hello, girls. Please have a seat. Please.
Thank you.
So, Edmund, I heard you turned these girls into meiosis experts. So, I brought a picture with me here today. And these are two homologous chromosomes. Would you mind explaining what this is?
Yes, this one, the yellow one, we're going to be showing in all of our diagrams, and this will be representing the father chromosomes, or the ones that the father gave to the haploid cells.
And this one will be representing the maternal cells, or the mom cells, and this is what they contribute.
Chromosomes.
Chromosomes, yeah. And this is what they contributed to the haploid cells.
My, how interesting. And is there like a certain phase for these cells? Would you explain?
Well, yes, we are going to be talking today about meiosis 1 and 2. So, the first part of meiosis 1 is interphase 1. And as you can see here, the black circle represents the cellular membrane. This green circle you see here represents the nuclear membrane. And the yellow one again is the dad's chromosomes, and this is the maternal chromosomes. And we just see here in this phase, as well as in mitosis, and the cell prepares for meiosis. And so centrioles duplicate, as you see here represented by the blue circles. Okay?
How interesting. And is there a second part to that?
Yes, there is a second phase, and it's called prophase 1. And as you see here, the black circle is the cell, and the blue part is the spindle fibers, which are beginning to form.
Mm-hmm.
And as you see here, the dad.
They are a homologous pair, right?
Yes. And as you see, the nucleus is beginning to dissolve.
If I must add, what is it that they're doing there?
They're synapsing, which is trading off their genes.
Genes, or
Traits.
DNA.
Yeah,
they're-
To one another.
Yes.
To one another, yes.
And actually, we have to mention that the homologous chromosomes we see here are they're homologous for various reasons. One being that they are the same length, and
They have the
Centromere?
centromere at the same location.
yes, and they have the gene for the same thing. For example, I don't know, eye color or hair.
Nose shape.
Yes. And now we move to the next. Miss, could you care to explain the next part?
Yes. What we have next.
Metaphase.
Where did you put it?
It's right there.
Metaphase.
I see it right here. Yes.
There it is.
Sorry. Okay.
So in metaphase, we see that the nuclear membrane is completely gone. And can you hold that for me, please?
Mhm.
And we see that the spindle fibers, which are presented in the blue here, have grabbed onto the chromosomes.
Homologous pairs, once again.
Yes. And also, if you can see in the mother and the father cell, you can see they have one another's genetic material.
Was that from the prophase?
Yes, it was from prophase 1 where they had synapsed and switched their genetic material.
On the tetrad, right?
Yes. And mainly also in metaphase 1, you see that they are aligned straight in the middle as a homologous pair, and then separated to the opposite end poles of the cell.
May I ask, what is the tetrad?
Give that to them. Let's test them on that.
Okay, well, a tetrad was formed in prophase 1, in LaQuisha's phase that she explained. And that one was the homologous pairs, when they synapsed, they made they formed a group of 4 chromatids.
Oh,
And they were put together.
I
Yes.
understand. I see.
Yes.
Okay, now we have anaphase 1.
Now, in anaphase 1, hold this please, they are also, again, pulled apart, and they're in the opposite poles of the cell. And the spindle fibers are now disappearing, getting smaller,
We're pulling them.
pulling apart. And there they are still a homologous pair, which they have split apart. Their genes have still been switched, and
They're preparing.
they are preparing to go to their next phase.
Which
Could you
is
pass me that one? I will explain this one. Okay. The last part of meiosis 1 is telophase 1, and sometimes they group telophase 1 and cytokinesis because it's just
The
The
same.
ending phase.
it's the ending phases. So in this one we see the
Cleavage?
I see some nuclear membranes.
Yes.
They seem to be forming.
Not only that, you can see as a cell has created a different shape.
Oh my.
Yes.
And that shape is called the cleavage furrow.
Yes, and what this does is it pinches the cytoplasm, and it will appear in the next parts we show you that it will make 2 daughter cells.
Mhm.
Now, the 2 daughter cells are diploid.
Yes.
What are
Diploid is doubled.
Oh.
so it means it has a
So like it's like D, like I
dot?
see.
Yeah,
Mhm.
exactly.
Hmm.
Yeah.
And that just means that because we humans in our cells, like mitosis, we have 46. So this means that we have 46 again, right?
Mhm.
Diploid.
So then does that mean they're separating chromosomes like half and half?
Well, yes, they will be known as haploid cells, or or they only have 23 chromosomes, but that's going to be in
Another
meiosis.
phase.
Two.
Oh, I see.
The next part. Yes, and the crossing over, it helps with the genetic diversity. So
It differentiates people. Like, we're not exactly like to our parents, and siblings like me and
Oh.
Shaniqua are nothing alike, and I'm of course hotter and way more attractive.
but I'm more
I
creative.
see.
Okay.
I just
What?
what?
No?
Let's
Okay.
get to hold
that's all right.
ourselves,
Okay.
ladies.
That's a hold of what we're saying.
Let's continue.
Mhm. Okay, now we'll move on to meiosis 2.
Here's your
Move this to this side. I will grab these.
Yeah.
Ready?
So in prophase 2, there, as you can see, there is 2 new cells, and they have the same.
Oh, hold it for you.
Yeah, thank you. The nuclear membrane is gone, as you can see, and the spindle fibers are beginning to form.
Right there.
Yes, that's right there. And the centrioles are on opposite sides,
Here.
you can see. And now it's beginning to go to the center to prepare for its next phase.
So this process is happening all over again.
Yes, it
Yes.
repeats.
Oh, my.
Yes, Professor, I mean, you're asking a lot of questions to us, but maybe the host knows something that care to explain anything, maybe.
Oh, me?
Yes.
Well, I remember back in my days when I was in high school, they told us 3 major differences between mitosis and meiosis because I often got very confused with them. So I learned in mitosis they use stomatic cells, while in meiosis they use gametes, also known as sex cells.
Mhm.
In mitosis, the number of chromosomes of the daughter cells would be exactly the same as the parent cells, while in meiosis, it's half. And mitosis, they reproduce asexually, while in meiosis, they reproduce sexually.
Okay.
It's getting hot in here.
You're very wise. Now,
Thank you.
how about we just show them what the second cellular division is?
Metaphase 2, please hold this.
This one, we just talked about prophase 2. So the second part, it would be metaphase 2. And this one, the chromosomes line up in the center, and we see the centrioles in the opposite sides. The spindle fiber is still there, and they are still the haploid cells that they are with 23 chromosomes.
If I may add,
Mhm.
you can see as also in metaphase 1, the spindle fibers grasp onto the
Centromere.
and are going to begin their next phase by pulling the homologous pairs apart.
Yes.
Mm.
And now we go into,
Bringing us to phase 2.
in this one,
As you can see, the sister chromatids are being pulled apart, as she said, by their centromere.
mirror.
And as you see, the spindle fibers are beginning to retract.
And actually, the major difference between anaphase 2 and anaphase 1 is in anaphase 1, parts that were being pulled apart were the homologous pair, but here it is the sister chromatids. As you see here, they are half of the original X-formed shape.
So they move to opposite ends of the cell.
Yes.
Oh.
And why do they do that, you may ask? Let's show them the last part of meiosis 2. Okay. Here we see very small 4. They're not drawn to scale. Okay. And so, explain them what happens here.
Oh, well, if the camera could get a little closer to see
Come here.
the not drawn to
details.
scale.
Come on.
Okay. So now we see the sister chromatids that were pulled apart, each in their own individual cell that once again did the cleavage furrow and broke apart. And now,
Cell division is complete with this last step of meiosis too.
thus creating the gametes, or sex
sex
cells.
cells.
And what type of sex cells are produced, you may ask? Here we go.
Well, I'll tell you about that one. There is our caged sperm and our cageous egg.
Haha. Okay, but you know, if we want to be scientific, it is sperm and egg.
Sperm and egg cell.
Yes, and it would be 4. And what kind of cell would it be? Diploid or haploid?
Diploid.
Diploid.
Really?
Really, Professor?
Am I wrong?
Really?
Prove me wrong then.
Professor.
It's a haploid.
Professor.
It is
What
haploid.
happens?
They have divided, don't you remember? They
Don't tell me.
They have divided.
I may be
May you
wrong.
guys explain what happens when these two cells meet?
Babies.
See,
Like
the
you
reason...
and me.
Make
Yes.
it more scientific. What happens is when the caged sperm or the sperm cell
With 23 chromosomes.
goes into the egg,
With
23 chromosomes.
they, well, obviously the
They will
sperm
be
loses
preparing
its
for
tail.
fertilization. So pretty much, we're creating the parts that we need to reproduce.
And create a
babies.
Life.
baby.
Or life. Okay, can we go back to here?
Yes.
We see the nuclear membrane reforming, and once again, it was reforming. The centrioles duplicated, and so now the cells will each be complete with their section.
Yes.
So this may be sperm cells reproducing here, or this might be some egg cells.
So I see that the result seems to be 4 haploid cells.
Yes.
Because they're half.
Half.
I see. How
Also
interesting.
What are they also known as? The 4 haploid cells? What's another? No, what's another name for
Oh,
them?
once again,
Gametes.
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 Yes. So, do we have? Can you bring back the
Cage.
yes. So
That's
yes.
male and male.
So the reason we need to go through meiosis is to make 4 haploid cells. And here's the 2 examples. Yes. And so, yes, that is what we have to present to you today.
Oh my, how intriguing. So that was the full process of meiosis we saw. So it's a pity, but we're running out of time.
We have to go. Do we
have to
really?
go.
Do we really?
Oh,
I don't want to go.
I'm sorry, students, but we have to go.
Oh.
Or do we?
Bum bum bum.
You're supposed to go out and then come back.
Okay. Okay. Well.
Well,
It's nice
It
meeting you.
was nice to meet you.
we
Nice
have to
to
go.
meet you,
Nice
Professor.
meeting you too.
Hope to see you later on in the
And
future.
Come on.
Have a nice
Wait,
day.
before we go,
We just want to give you a little special something.
we need a song.
A
We
dance.
need our exit song.
You know?